Hi everybody. In this video, we are going to discuss about the special modes of reproduction. Special modes of reproduction that is taking place in the flowering plants. So we know that in the process of reproduction reproduction is of two types, asexual reproduction and sexual reproduction. But here the plants, the angiospermic plants will undergo two types, as we said, sexual and asexual, but here we are going to discuss about the other special modes of reproduction. That is apomixis and polyembryony. So in today's class we are going to discuss about the apomixis. So what this term represents? What do you mean by this term apomixis? Here in the very beginning, let us try to understand what is apomixis and polyembryony. Here, apomixis means when the seeds are produced without fertilization. We are calling it as the apomixis. That is the basic point we have to remember when we talk about the apomixis. Then here, what is polyembryony? Poly means many. Embryony means embryo. Within one seed, there may be more than one embryo present. So that's why, see, these two are not so common. It occurs in some angiospermic plants. So here, that's why we are saying that these are special modes of reproduction. So here, let us now understand very clearly about apomixis. The term apomixis is given by a scientist known as Winkler. He is the first person who used this term known as apomixis. So the term apomixis is given by Winkler. So as we said here, apomixis means what? The seeds are formed without fertilization. We know that the fertilization, the end product of fertilization is what? Seed. The end product of fertilization is seed. But here we are coming across the special mode of reproduction saying that seeds are formed even without fertilization. That is the interesting point we have to keep in mind. Seeds are formed without fertilization. So here the first point that we have to remember when we discuss about apomixis is it is a mode of reproduction. So in this there is no gametic fusion. In sexual reproduction, what do we say? That there will be a male gamete. And which will fuse with the female gamete. Male gamete will fuse with the female gamete. So when these two fuse together, it is giving rise to what? It is giving rise to the zygote. Which is a single cell and diploid. Diploid zygote we see. And this zygote now develops into embryo. And this embryo is present where? It is present in the seed. So here this process is what? Sexual reproduction. But here what we are saying? It is not sexual reproduction. Now we are giving one new name called as what? Apomixis. In this, even though we are saying it is a mode of reproduction, what is not taking place? There is no gametic fusion. There is no male gamete fusing with the female gamete. And that's why when there is no fusion of male gamete with the female gamete, then obviously there will be no formation of what? Zygote. Zygote will not be formed. Here only when male gamete fuses with the female gamete, what is formed? Zygote is formed. But here what we are saying is that when there is no gametic fusion, there is no chance of what? There is no chance of zygote. Because there is no fusion of male gamete with the female gamete. So here there is no formation of zygote. But still, what we are calling it? We are calling it as a mode of reproduction. It is a mode of reproduction. And in this process, it involves only mitotic divisions. Which means it is always maintaining the same number of chromosomes. And there is no reductional division in the number of chromosomes. Means in simple, no meiosis. What is not taking place? No meiosis process is taking place. Reductional division, half the number of chromosomes. So here, for the formation of the male gamete and for the female gamete, compulsorily what is the process that occurs? It is meiosis actually. Meiosis will be taking place. So here, in the process of apomixis, it will only undergo mitotic divisions and no meiosis will take place. And here, it is because mitotic division, same number of chromosomes are carried. And please remember, the newly formed individuals through a process called as what? Apomixis are similar to the parent. They are exactly the same as the parent. What is exactly same as the parent? The individuals that are produced by a process called as what? Apomixis are exactly resembling the parent. They are genetically similar to the parent. So once we say that they are genetically similar to the parent, okay, what are they called as? We are calling them as what? Clone. So genetically similar individuals obtained from the same parent means what? Single parent producing the young ones, and that young one will be exactly resembling the parent. So when it is exactly resembling the parent, it means that all the characteristics that are there present in the parent are now transferred to the young individual or new individual, which is exactly the same. And once they grow up, when this new individual grows, when you are keeping the parent and the young one offspring, they will be exactly the same and they do not differ in any characteristic. All the characteristics are same. So such genetically similar individuals obtained from the same parent are called as what? We are calling it as clone. And the cells in the clone, say suppose these are all the cells which are just identical. All of them are alike. They are same in all the characteristics, either it is in the shape or in the color or any genetic composition, everything they are exactly the same. So when they are all same, we are calling all these cells as what? Present in this young one clone. And each cell in the clone, one individual cell we are taking up. If this is one individual cell, that individual cell will be called as what? Ramete. It is called as the ramete, which is a very important term. Okay, which is a very important term you have to keep in mind. So individual cells in a clone is called as what? Ramete. Individual cells in a clone is called as ramete. Now, the organisms that reproduce, okay, apomixis. You are now taking one organism. It is reproducing the young ones through apomixis process. Such organisms are called as what? Apomix. So if you are seeing one plant which is producing the seeds without fertilization, that plant will be called as what now? Apomix we are calling it as. Okay, we are calling it as apomix. And now if you are clearly observing, all these characteristics are observed or identified in the asexual reproduction. So what is asexual reproduction? Asexual reproduction means producing the young ones of their own type, in which only single parent is involved. Where there is no gamete fusion. What we say that is commonly as what? Asexual reproduction. So in the same way here, the point that you have to keep in mind is that apomixis is nothing but it is similar to asexual reproduction. Okay, it is similar to asexual reproduction, or it is a method of asexual reproduction only. Apomixis is a method of what? Asexual reproduction only. As we say now, we can now say that asexual reproduction can be vegetative propagation or vegetative reproduction and apomixis. So asexual reproduction is of two types, we can say now. Asexual reproduction is of two types. What are those? Vegetative propagation or vegetative reproduction. That is the first one. And the second one we can say it as what? Apomixis. So apomixis is nothing but what? A type of asexual reproduction. Now when you see this in plants, okay, in plants, Asexual reproduction, that is nothing but what now? When you are saying asexual reproduction, what is that now? We are talking about what? Apomixis. Just now we said apomixis is nothing but what? A type of asexual reproduction. So let me say here, in plants, asexual reproduction means what? We have to keep in mind whenever you are coming across here the word apomixis. Okay, apomixis. So here in plants, asexual reproduction means apomixis mimics what? Sexual reproduction. It mimics sexual reproduction but produce seeds without fertilization. Means what? Apomixis is exactly like what? The sexual reproduction. Exactly like, or it is mimicking the sexual reproduction in what? In producing the seeds. Because what we said, in sexual reproduction, what will be produced? Seeds will be produced. So what we are saying here, even asexual reproduction mimics the sexual reproduction. But here, it is producing the seeds without fertilization. But in sexual reproduction, it produces the seeds through fertilization. In sexual reproduction, it produces the seeds through fertilization. But here, what we are saying, even in asexual reproduction, seeds are produced, but without fertilization. So that's why we are saying, if the question is asked, what is apomixis? The direct thing that you have to remember, you can say is, it is asexual reproduction which mimics sexual reproduction. Asexual reproduction which mimics sexual reproduction, but produce seeds without fertilization. Okay, so otherwise we can simply say asexual reproduction, sexual reproduction. In asexual reproduction, seeds are produced. Sexual reproduction, seeds are produced. In asexual reproduction, seeds are produced without fertilization. But whereas in sexual reproduction, seeds are produced through fertilization. That is the main difference we have to keep in mind. And now here the common thing is what? Production of seeds. And the production of seeds through apomixis, that is asexual reproduction, is very common. In some of the best examples is in the members of Asteraceae. In the members of Asteraceae, we can see this process called as asexual reproduction. What do you mean by now here in this topic? What is the point we have to remember when we talk about asexual reproduction? Apomixis. So apomixis is very clearly observed in the members of Asteraceae and grasses. In the members of Asteraceae and grasses, we can see the process of apomixis. So in this, as we are continuing to learn this, remember now apomixis is nothing but production of seeds without fertilization. Or we can say that it is a type of asexual reproduction which mimics sexual reproduction but produces seeds without fertilization. So here we should remember that asexual reproduction can now, in a broad sense, can be said as what? It is of two types, agamospermy and vegetative propagation or vegetative reproduction. Asexual reproduction is agamospermy or vegetative propagation or vegetative reproduction. It is of two types we say. Here already what we said, apomixis is a type of what? Asexual reproduction. What we said when we are talking about apomixis, seeds formed without fertilization. Okay, is called as what? Apomixis we said. Apomixis. So here let us be very careful when we are understanding this. Seeds formed without fertilization means here we also should keep what is there present in the seed. Seed compulsorily, seeds are formed means compulsorily what should be there inside? An embryo should be there. But what we are saying, seeds are formed without fertilization. So here the word or the term apomixis is for the seed. What is there present inside the seed? It is the embryo. So formation of embryo, without fertilization and meiotic division is called as what? agamospermy. Agamospermy. So here we are coming across a new word now. New word, that is agamospermy. Agamospermy. Agamospermy means what now? It is a method in which embryo is formed without fertilization. But here, where is the embryo present? Where is the embryo present? The embryo is present in the seed. Okay. Seed here, if you are looking at a seed, is having what now? The embryo. The seed is having the embryo. So, if the embryo is formed without fertilization, we are calling it as agamospermy. And if the seed is formed without fertilization, we are calling it as apomixis. Almost both are the same. Seed consists of what? The embryo. Seed consists of embryo. Seed is formed without fertilization, and that seed is containing the embryo without fertilization. So, embryo that is formed without fertilization, we are using the word agamospermy. Agamospermy. So, agamospermy is a method of what? Asexual reproduction. So, embryos, in this method, embryo is formed without fertilization and meiotic division. Meiotic division. No meiosis will be taking place. No reductional division will be taking place. And these plants belonging to this category propagated through what? The seeds. Those embryos which are formed without fertilization and meiotic division are propagated by what? Through the seeds. But embryo formation does not involve what? Meiosis and syngamy. Syngamy means what? The same, fertilization. Okay, fertilization. So both, it is giving the same meaning here. So we have to remember that the seeds, formation of seeds without fertilization is apomixis, and formation of embryo without fertilization and meiotic division is agamospermy. Agamospermy. So now we are going to discuss about agamospermy. Agamospermy. Agamospermy means what? It is the formation of embryo. It is the formation of embryo without syngamy, means without fertilization and meiotic division. Embryo is formed without fertilization and without meiotic division. That process we are calling it as what? Agamospermy. So it can occur in various methods. Such as apospory, diplospory, and adventive embryony. Apospory, diplospory, and adventive embryony. In all these three ways, the embryo will be formed without fertilization and meiotic division, which we are calling it as agamospermy. So we will study one by one in very detail. We are going to discuss now. Now, the first one under agamospermy is apospory. Apospory. So our main theme is what? We are talking about how the embryo is formed without syngamy and without meiosis. So here the first one is apospory. Apospory means formation of gametophyte. Directly from sporophyte. Here gametophyte is what? Haploid. It will be haploid. Whereas sporophyte is what? It is diploid, isn't it? Generally the gametophyte will be haploid and sporophyte will be diploid. But here the conditions are different and the process is different under apospory. Formation of gametophyte directly from sporophyte without meiosis, which means now the sporophyte, which is what? Diploid. Please remember, the sporophyte which is diploid will now produce the gametophyte. Which is diploid. That's what. But what we said actually, what is the process? A diploid cell will undergo meiosis to produce the gametophyte. But here meiosis is not taking place. So that's why sporophyte without meiosis, it is producing what? Gametophyte. Which means now it is also diploid only. It is also diploid only. So that process we are calling it as what? Apospory. Formation of gametophyte directly from sporophyte without meiosis is called as apospory. For the first time, this process, apospory, is discovered by a scientist known as Rosenberg. Okay, by the scientist known as Rosenberg in the Heratium plant. In the Heratium plant, he discovered the process of apospory. What is that apospory? Apospory means the sporophyte, diploid cell, without undergoing meiosis, it produces the gametophyte, which is also a diploid. So that is being discovered by Rosenberg in Heratium plant. Please remember this, Heratium plant. So here, now the embryo sac. What do you mean by this gametophyte? It is nothing but the female gametophyte. It is the female gametophyte. Embryo sac or female gametophyte. Female gametophyte is directly formed from any diploid cell of the sporophyte. Now we are talking. Now if you are looking at this diagram, okay, if you are looking at this diagram, this is an embryo sac. This is an embryo sac. So this embryo sac is actually formed by what process? Meiosis. It is formed through meiosis process. But here what we are saying is that the embryo sac is formed without undergoing meiosis. That embryo sac is nothing but female gametophyte. So here, this is the embryo sac. Otherwise, we are calling it as what? Female gametophyte, which is now, this embryo sac is diploid. It is diploid. Please remember that. It should be actually haploid, but it is now diploid. How or why it is diploid means it is directly formed from the nucellar cells. One of the nucellar cells will directly develop into what? Embryo sac without undergoing meiosis. Or otherwise integuments of the ovule. Now as we have seen, okay, say suppose if this is the ovule, okay, if this is the ovule, all these cells out of the embryo sac, what are these cells that are there outside? We call these as the nucellar cells. And what are these? We call them as integuments. So any one cell of the nucellus or of the integuments which are diploid directly develops into an embryo sac, female gametophyte. Directly develops into an embryo sac, which is called as female gametophyte. Here, if you are taking a nucellar cell, this nucellar cell, let me say nucellar cell, it is developing into the embryo sac, female gametophyte, embryo sac. Okay, embryo sac. And the embryo sac, okay, will be containing what? It will be containing the antipodal cells. Okay, it will be containing the egg cell. And it also contains what? Synergids. Now here is this structure. This is the embryo sac. It is containing synergids. It is containing the two synergids. It is containing the egg. Here is the central cell. Of course, this is what? The central cell. And it is having antipodal cells. Now, these are the basic things. Or otherwise, we can say even the central cell. Okay, central cell. So, what we said now? When a nucellar cell without meiosis, develops into embryo sac. It is diploid embryo sac now. Formed from what? Diploid nucellar cell, embryo sac. And this embryo sac is also having the antipodal cells which are diploid, egg cell which is also a diploid, synergids which is also a diploid. Mainly these three, okay, these three, antipodal cells, egg cell and synergids, okay, they are all what? They are diploid. They are diploid. Antipodal, egg cell and synergids, all these three are diploid. Now what we said, embryo is formed without fertilization and without meiosis. So here is without meiosis, okay. Now we will be seeing that embryo will be formed. Now here, nucellar cell without meiosis forming the embryo sac, and this embryo sac is containing the antipodal cells, diploid, which are all diploid, egg which is diploid, and synergids which are diploid. And the most common thing is that egg, okay. Here what we say is that egg cell, okay, where is the egg cell now? Present in the embryo sac. This egg cell which is diploid, now without fertilization, okay, without fertilization, it will develop into embryo. It will develop into embryo, okay. It will develop into embryo. Is it clear? So here what we should remember is that apospory means the sporophytic cells like nucellars or the integments will, without meiosis, they form the embryo sac, and this embryo sac is containing the cells which are having the diploid number of cells or chromosomes called as antipodal cells, and egg cell and synergids. Now egg cell without fertilization, it is directly developing into embryo, So here, whenever in this process, whenever the egg is developed into embryo, we call it as diploid parthenogenesis. Remember this. We call it as what? Diploid parthenogenesis. Please remember, this word is an important term. Diploid parthenogenesis. Diploid parthenogenesis means what? When the egg cell without fertilization developed into embryo, we call it as what? Diploid parthenogenesis. This is within what? The apospory only, within the apospory. And please remember now, apart from the egg cell, which is very common to develop into embryo, there are chances that antipodal cells and also synergids will also develop into what? Embryo. There are chances. Each antipodal cell can develop into a new embryo. Each synergid can develop into a new embryo without fertilization. So, except the egg cell, if the antipodal cells, okay, if these antipodal cells and the synergids, okay, these antipodal cells and the synergids, if they are developing into embryo, we call it as apogamy, diploid apogamy. So remember that, we have to remember that when the antipodal cells, okay, when the antipodal cells or synergids, when they develop into embryo, this is also without what? Without fertilization. We call this process as what? Diploid apogamy. Please remember, diploid parthenogenesis means what? When the egg cell is developing into embryo. Apart from egg cell, except egg cell, if the antipodal cells and the synergids are developing into the embryo, we call it as what? Diploid apogamy. Okay, this process can be called as diploid apogamy. Diploid apogamy we are calling it as. So here we came across two words, diploid parthenogenesis and diploid apogamy, under what? Apospory. Apospory means what? When a sporophytic cell develops into embryo sac or gametophyte, it is called as apospory. And that embryo sac consists of what? Egg. It consists of synergids. It consists of the antipodal cells. If the egg is developing into embryo without fertilization, diploid parthenogenesis. If the antipodal cells or the synergids are developing into embryo, we call it as diploid apogamy. We are calling it as diploid apogamy. So these are some points we have to remember when we talk about what? The apospory. So what are the best examples of apospory? Okay, apospory, the best examples are ranunculus, heratium and rubus. Okay, and rubus. These are the best examples which show apospory. Next one we are going to discuss is about diplospory. Okay, diplospory, in which the embryo is formed without fertilization and without meiosis. Without meiosis. So the next one is diplospory. Here the megaspore mother cell, which is diploid. And you may get doubt, what is this megaspore mother cell and where it is present? This megaspore mother cell will be developing into what? Embryo sac. Then this megaspore mother cell is present in the ovule. Which is a very familiar diagram for all of us now. The structure of ovule. Here, all these cells are what? The nucellus cells. All these cells present in the ovule in the beginning, they are called as what? Nucellus cells. Nucellus cells. One of the nucellus cell in the ovule. What is the structure we said? Ovule. Okay, this is the ovule. One of the nucellus cell at the micropyle end. Let us say that this is the micropyle. Okay, micropyle. So at the micropyle end, one of the nucellus cell, which is what? Diploid, will undergo. Actually, it will undergo meiosis. It will undergo meiosis and produces 4 cells. Okay, and produces 4 cells. These are all, okay, the megaspore cells we call them as, and from which they are formed is called as what? Megaspore mother cell. That one single nucellus cell which is dividing through meiosis will be called as what? Megaspore mother cell, which is diploid. But at the end, the diploid nature will be disappearing through the meiosis and produces all these cells which are what? Haploid. Which are haploid. And one of that, let us say the terminal one, okay, this one will be developing actually into what now? The embryo sac. The embryo sac. This is the structure which will be formed from that one single cell. Okay. All this process, okay, this complete process, what we call it as megasporogenesis. Okay, megasporogenesis we call it as megasporogenesis. So here what we are saying is that one megaspore mother cell, will not undergo megasporogenesis. Means what? No meiosis. Meiosis will not take place. Only this megaspore mother cell without meiosis directly, that single cell which is diploid, directly develops into what? Now embryo sac. So here is the embryo sac which is diploid. Diploid embryo sac. Produced from this diploid embryo sac is produced from what? Diploid megaspore mother cell. Megaspore mother cell without meiosis. And this embryo sac will be having the antipodal cells which are all diploid only. Even the egg is also diploid. Diploid egg cell. Okay, diploid egg cell. And even the synergids are also diploid only. Are diploid. So here we have to remember that the megaspore mother cell which is diploid, without meiosis, it is directly developing into an embryo sac having antipodal cells, egg cells and synergids. All these are there. Now the same thing as we have seen in apospory. When the egg cell, okay, when the egg cell develops into what now? Embryo. When the egg cell develops into embryo. So here, if you are looking at the embryo sac, it is consisting of what? The antipodal cells, the egg and also the synergids. Here, these are the 3. So here, whenever the egg cell, okay, diploid egg cell, please remember this is what? Diploid egg cell. Actually egg will be haploid, but here it is diploid. When the diploid egg cell without fertilization, okay, without fertilization, when it is developing into what now? The embryo. Okay, when it is developing into embryo, okay, we are calling this actually, which is coming under what? Diplospory. This we call it as again the same word called as what? Diploid parthenogenesis. Diploid parthenogenesis we are calling it as diploid parthenogenesis. But if it is the same like antipodal cells and synergids, okay, either antipodal cells, any antipodal cell, or let us say synergids, okay, synergids, when they without fertilization, okay, when they without fertilization develops into what? Embryo, okay, when they develop into embryo. So here is again the embryo that is formed from what? The antipodal cells or the synergids. It is not from the egg. Actually, the embryo will be formed from the egg. But here what we are saying, the embryo is formed from the antipodal cells. Okay, antipodal cells and or otherwise the synergids. The synergids will be developing into embryo. In such case, we will be using the same word called as what? Diploid epogamy. We call it as diploid epogamy. We are calling it as. And very important point is that we should always remember it is the egg that will be developing into embryo. And if not egg, which is the cell develops into embryo? Is it the antipodal cell or the synergids? Which one will be given more preference to develop into an embryo means? Please remember the most common, okay, cell that develops into embryo is the synergids. Please remember, okay, synergids will be having more chances, okay, synergids will be having more chances to develop into what? Embryo. So that those synergids are what? Those synergid cells are what? Diploid only. Or it is antipodal or synergids, they are diploid cells developing into embryo, which we call it as what? Diploid epogamy. Diploid epogamy. The plants which are showing diplospory are Parthenium. The best examples are Parthenium and Taraxacum. Taraxacum, which belongs to Asteraceae. Which belongs to Asteraceae. In these two, it will be showing what? Diplospory. So here the next one is adventive embryony. Adventive embryony, which is also called as sporophytic budding. This is the last type. Okay, this is the last type of agamospermy. Adventive embryony or otherwise called as sporophytic budding. So in this, embryo is formed from any diploid cell or like diploid cell of what? Maybe the cells of nucellus or the integuments. Cells of the nucellus or integuments which are outside of the embryo cell. This is the embryo cell. So here, say here, if this is the, okay, if this is the embryo sac, if this is the embryo sac, outside what is there? The nucellus cells, and these are what? Integuments. So this we can say that these are the nucellus, and this is what? The integuments of the ovule. Integuments of the ovule. So here the point is that embryo, not embryo sac. Embryo is directly formed from the diploid cell. Means one of the nucellus cell here, nucellar cell directly develops into what now? It develops directly into embryo. So remember that this embryo is formed outside the embryo sac. Outside the embryo sac. Or even the same, if it is integuments also, any cell of the integument, can directly develop into what? The embryo. Directly develops into embryo. So that's why embryo is formed from any diploid cell. So nucellar cell or integuments, they are what? Diploid cells which directly develop into the embryo. So here what we have to remember, this nucellar cell or integument is behaving like what? Zygote. So here zygote, so let us say that this nucellar cell is nothing but what? So zygote. So zygote now develops into what? Embryo. What is this zygote? It is nothing but either as it may be a nucellus cell or integument. So these are behaving like as zygote, and they form as what? Embryo outside the embryo sac. So that's what we are saying. Diploid cell. What could be the diploid cell? Either it is nucellus or integuments. Behaves like zygote and this zygote, nothing but nucellus or integument, will develop into what? Embryo. Develops into embryo. So here, please remember, the embryo that is formed outside the embryo sac will be pushed inside into the embryo sac. Into the embryo sac. Because the zygote or the embryo is pushed into the embryo sac, that's why we are calling it as what? Sporophytic budding. Sporophytic means what? It is diploid cell. It is diploid cell. What is that diploid cell? Nucellus or integument. These are behaving as what? The zygote. Then zygote will grow into what? Embryo. Embryo. So this embryo is now what? It is diploid, made up of diploid cells only. But all the way embryo is formed from the nucellus or integuments outside the embryo sac. Outside the embryo sac. And this embryo is pushed inside into the embryo sac. Because it is pushed like a small bud inside into the embryo sac, we are calling it as what? Sporophytic budding. Sporophytic budding. So when the embryo is formed outside the embryo sac and pushed inside into the embryo sac, we call it as what? Sporophytic budding. And here, if you are looking at the examples showing adventive embryony, the best examples are the nucellus cells which develops directly into embryo is seen in citrus. Mango, opensia and onion. In all these, the adventive embryo which is formed outside the embryo sac is pushed into the embryo sac, and from which cell means it is from the nucellus. Likewise, the integuments which develop into zygote and thereby into embryo is seen in Spiranthes australis. Spiranthes australis. So these are some examples we have to remember when we talk about adventive embryony. Adventive embryony. So after learning all these, what is the advantage now? After learning all these three types, epospory, diplospory and adventive embryony, what are the advantages for us or for the farmer? Is there any advantage for the farmer? We are going to look at now. So after studying about the agamospermy, what is the advantage for a farmer? Because here there are many problems that are faced by the farmers. What are those problems? Hybrid seeds have to be produced year after year. Now hybrids means what? The offspring, okay, the seeds that are produced from a plant are called as hybrids, having the selected characteristics. But when you take those plants, suppose there are two plants, when you pollinate them and get these seeds, from this crop, okay, these seeds will be having the characteristics of both the parents. And these seeds, when you sow them, okay, when you put them into the soil, when they start growing, they may be pollinated by other agents, and that's why what happens, there will be segregation of characteristics. Other parent characteristics also will be present, and when these now are crossed with some other, now they will be carrying different characteristics, which means hybrids or the offspring will not carry the same characteristics year and year. So what we are saying here, seeds have to be produced year after year. Means what? The seeds with the selected characteristics have to be produced year after year. But that is not possible. Because of what? Because why? When the hybrids are sown, progeny will segregate. The characteristics were separated. The same characteristics will not be there. Progeny will segregate and do not maintain the same characters. They do not maintain the same characteristics which are desired by the farmer. And to produce the hybrids with desired characteristics, it is very costly. These are some of the problems. Now, knowing these problems, now we can overcome these problems if the seeds are made apomictic. Okay, if the seeds are made apomictic, means what? Now you know apomixis means what? Seeds that are formed without fertilization. If the seed is formed without fertilization means what? It will be carrying the same characteristics. Diploid cell is changing into an embryo. So it will be carrying the same characteristics year after year. So now the research is being done in the laboratories to know the selected genes which are responsible for carrying these characteristics from one generation to another generation, and now the scientists are trying to identify the gene and place that gene in the hybrids so that the characteristics will be there present year after year. The characteristics will be there present year after year. If the scientists are successful in researching in the laboratories about how to identify the gene which is responsible for apomixis, and if they can transfer this gene into the hybrids where there will be no segregation at all now, then the farmers will no need to spend a lot of expenditure on their field to cultivate the crop with desired or selected characteristics. Because in apomixis, what we said, there is no fertilization. There is no seeds are formed without fertilization and without meiosis, we said. Means same characteristics are carried out. So that's why they are doing research on this, so that they can produce the seeds which are apomixed. So these are the points which you keep in mind when we talk about the apomixis. Thank you.